Hello, I would like to welcome you to this exciting webinar that we have today. It will be on implementing the ISBT 128 standard in India. We'll be touching on regulatory alignment, traceability, and practical experience. I'd like to introduce myself. I am Robin White. I am the Deputy Technical Director here at ICCBA. We're grateful for this collaboration with Kasturba Medical College, Manipal, and we look forward to an engaging and insightful session. I'd like, on behalf of the webinar team, to extend a warm welcome to both our distinguished chairpersons, Dr. Deepika Channa, additional professor in the Department of Immunohematology and Blood Transfusion at the Kasturba Medical College, Manipal. Joining her is Dr. Debashish Gupta, President of the Indian Society of Transfusion Medicine and Professor and Head of the Department of Immunohematology and Blood Transfusion at the National Institute of Medical Sciences and Research. Our chairpersons will have the pleasure of introducing each of our speakers and facilitating the discussions throughout the webinar. Before we get started, I would like to give a brief background on ICCBA and the ISBT 128 standard. So, established in 1994, ICCBA is the international nonprofit, non-state actor supported by user fees, tasked with developing and maintaining the ISBT 128 standard for medical products of human origin, or MPHO. We cover the terminology, globally unique identification, labeling, and electronic data exchange for medical products of human origin. We have a small staff of 17 people, and we rely heavily on our technical advisory groups, which are made up of volunteer experts from around the world. We also closely work with other standards development organizations, such as HL7, as well as the standards coordinating body, to provide solutions for challenges in the healthcare information technology field. And now a little bit about the origins of the ISBT 128 standard. Back in the 1990s, some problems, or we could call them opportunities for improvement, started occurring. First, the previous identification methods were becoming no longer adequate for blood product labeling needs. For example, Codabar was running out of codes. There was no system for globally unique identification, and increasingly, blood products were being exchanged across regions. It was also recognized that a system for traceability was essential for global hemovigilance and biovigilance. And to solve these issues, stakeholders from around the world, including organizations like ISBT, which is the International Society of Blood Transfusion, and AABB, the Association for the Advancement of Blood and Biotherapies, developed a global standard to support identification and traceability, and that's the ISBT 128 standard, which ICCBA manages. Like I said, we are heavily dependent upon our technical advisory groups, the experts in those technical advisory groups. ICCBA currently has a total of 10 tags, technical advisory groups, but we will be combining our 3 regional blood tags into one international blood tag later this year. The individual chairpersons from each tag sit on the standards committee along with other experts to provide final approval for the standards. And I'm very happy to say that in the past 30 years, over 600 volunteers have contributed to developing and maintaining ISBT 128. And now on to our webinar. Throughout the webinar, we encourage you to submit questions using the Q&A feature located in the Zoom toolbar. Depending on your view, you may need to select the More button, which is the 3 little dots, to access the Q&A feature. Please note that we will not be monitoring the chat feature. We will be monitoring the Q&A feature. So with that, I'd like to invite Dr. Deepika Channa to the webinar stage to introduce our first speaker. Thank you, Dr. Channa.
Thank you, Dr. Robin. It is a privilege for me to introduce our first speaker for today's session, that is Professor Dr. Shamee Shastry, who is a professor in the Department of Immunohematology and Blood Transfusion at the Kasturba Medical College, Manipal Academy of Higher Education, Manipal, Karnataka, India. She has 18 years of research experience and she has completed her MD in Transfusion Medicine from Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow. Dr. Shastry is a researcher in transfusion medicine with over 130 Scopus indexed publications and a H index of 17. She coordinates the Center of Excellence in Immunohematology at the Institute and also serves as a secretary of the Asian Association of Transfusion Medicine India chapter. Her accolades include the Harold Gensen Fellowship by the International Society of Blood Transfusion, the Young Scientist Award by the Indian Society of Transfusion Medicine, and the Indian Society of Blood Transfusion and Immunohematology. She also collaborates with the Transmit Africa project, which is an intercontinental initiative to improve the blood transfusion safety, which is funded by the Erasmus Plus European, led by the Autonomous University of the Barcelona, where the Manipal Academy of Higher Education is an associate partner and is working in collaboration with the Asian Association of Transfusion Medicine. To her credit, she also has been the pioneer in establishing the South India's first rare donor registry and also in identifying the P null phenotype for the first time in India. These are just career highlights which are only selected points, and Madam is an inspiring mentor for many students as well. Her vision is mainly focused on improving the transfusion service in India by applying the scientific advances into practice. With this brief introduction, I would like to welcome Dr. Shastri for her session. Over to you, Madam.
Good morning, everyone. Thank you, Team ICCBBA, for this opportunity. So, we transfusion medicine personnel spend a significant amount of time and resources in perfecting the blood inside the bag, doing serology testing and storage. And across published data, many of the blood component errors trace back to something far simpler that's written on the outside the bag, a label. So over next 15 minutes, I want to walk you through the regulatory anatomy of labeling in India. I will move from the matter that why the label is important through the definitions and the regulatory framework in India. Let's start with the clinical context. So we need to treat the label as the first safety system, not as a paperwork. It's the first check and the last safety barrier. We know that 35% of the serious hazards are traced to errors in the component identification or labeling. Even India's hemovigilance program increasingly highlights the documentation that's related to labeling as the root cause. And before discussing the blood component labeling in detail, it is important to recognize why it matters. This study on errors across the transfusion chain provides a powerful message. What is particularly striking is that the blood component labeling errors accounting for the 31% of all the lab near miss events. And here we have a larger study that demonstrates the true scale of the problem. The Pareto chart analysis here reveals the important finding. The majority of the errors originated from just a few recurring causes that included labeling related issues contributing to the largest share of incidents. So labeling errors rarely have a single cause. They are usually the final outcome of multiple weaknesses across the system. As shown in this fishbone analysis, the errors can arise from people, processes, equipments, materials, information management, and the work environment. So if we are able to reduce or eliminate labeling errors, what would be the impact on overall transfusion errors? As shown in this waterfall chart, there can be about 30 to 35% reduction in the error rate. So the message is simple. Every improvement in labeling translates directly into the improved patient safety. So with this clinical background, now let us look into the key definitions and the regulatory framework at India. This is the legal backbone, and we all know that the apex body is CDSCO, and Section 3B of Drug and Cosmetic Act is what makes blood a drug in Indian law. And Schedule F Parts 12 provides the blood center standards, and in addition, we have some of the supporting guidelines and the standards by National Blood Transfusion Council as well as NABH, and also released by DGHS, the technical manual. Cellular therapy products have multi-authority regulatory architecture regulated primarily by DCGI, and the RCGM, the Review Committee of the Genetic Manipulation, the Institutional Biosafety Committee, and DBT also have a major, the significant role to play. Coming to the key definitions, what is a label? It's nothing but an inscription affixed to a unit for identification. And labeling is the process applying a highly regulated, standardized label to a blood bag or the container. So each blood center shall maintain a policy document and a standard operating procedure, and also a master file of all the labels in the prescribed format as mandated by drug and cosmetic rules. And this slide summarizes the process flow for labeling and the blood components from the donor bag selection through the final compatibility testing labeling. Regarding the label requirements, the Schedule F of Drug and Cosmetic Act specifies 14 mandatory particulars, and I'll go through each cluster briefly. Point 1 to 4 cover the identification and administration particulars. The proper name of the product should be there, the blood center name and address, the license number, and the serial number, that's nothing but the unique donor identification number, shall be on the label. Point 5 to 8 about the date, the color coding as per the ABO blood group, blue for O, yellow for A, pink for B, and white for AB, gives a rapid visual check before anyone reads the text, and also the report of the infectious marker testing that we do. The volume composition and the information help ensure the correct therapeutic dose, while the storage temperature confirms the product quality and the safety have been maintained throughout the supply chain. Point 11 mandates the use of transfusion set with inline filter, and the point 12 about the compatibility label. However, the DGHS technical manual 3rd edition gives us the fullest field level guidance for the compatibility label, mentioning the patient's full name, hospital identification number, the unit number, blood group, etc. And the point 13 is a clinical judgment embedded in the regulation. It's about the visual inspection of the unit. And point 14 is about indicating the donor classification that must be not less prominent than any the product name on the label. So here we have the latest Indian Pharmacopoeia monograph list, and note how granular the platelet nomenclature has become now. Earlier we used to just mention as platelet concentrate. However, now we can also add on the preparation techniques as well. So now it's time to update or correct the existing labels and SOPs as per the current monograph. A few additional practical details are given by the DGHS technical manual as well as standards by NBTC. And one such example is about the PRBC that can be prepared from the blood bags containing up to 35% lower than the total volume. These PRBC are labeled as low volume units and can be used for transfusion. In addition, regarding the date of expiry, Drug and Cosmetic Act just mentioned it has to be noted on the label. However, the technical manual has clearly given out the method of calculation of date of expiry. The date of expiry of the blood component is calculated considering the day of collection as day 0. In addition, whenever we modify the blood components, the details must go onto the label. For example, pooled platelets. So each pool should be labeled with volume, the blood group, and the number of units in the pool. Further, when we irradiate, leukodeplete, or wash the components, we need to add on the details. The expiry gets modified when we irradiate or wash the components, and similarly, the leukodepleted product. And for autologous blood products, we have to mention clearly it's for autologous use only, and the donor signature should be on the label. In addition, some of the important additional points mentioned in NBTC standards that is not mentioned in Drug and Cosmetic Act. This is to have one such example is to have a acceptance criteria for label, because label is a critical material of blood center, and also about the quality of the label has to be firmly attached to the blood components, because cold chain condensation genuinely degrades these, it affects the ink or the adhesive nature. Further, NBTC explicitly recommends the use of barcode or RFID traceability for real-time visibility. So if your center is planning a barcode upgrade, and then this is the clause for your regulatory justification for it. NBTC standards also provides the detailed guidance on the transfusion instructions that should be included. In Drug and Cosmetic Act, it is just mentioned that the instructions have to be there, whereas the detailed instructions is given by the NBTC. So, although regulations define what must appear on the label, it is not standardized how the label are to be designed or generated. As a result, significant variability exists, and across the blood centers, this creates potential gaps. The variability begins with the fundamentals such as label materials. It can be plain paper or adhesive. It varies from center to center. It may be a pre-printed stock label or blank fill letter, and it may be a synthetic or polyester in nature, or pasted on the bag, or some centers even use it as a tie tag. And regarding the data entry, it may be handwritten or fully manual system, whereas some centers have adopted semi-automated pre-printed, and in few centers they have adopted ISBT 128 or barcode driven system. Even the timing of application varies. It may be immediately after component preparation, or after cross-matching or compatibility testing, or it is stuck on the unit only at the point of issue. Even the layout has no fixed quadrant convention. Every center's label essentially looks different. The ID numbering system are center specific. So the challenge is not the lack of labeling, it's the lack of uniformity. Standardization remains the key for the safer and efficient transmission practice in this aspect. So this is one such example where though there is a standard color coding, however the shade may vary. So the pink or yellow, the blue shade that we use varies from center to center, from vendor to vendor. Even the handwritten the information that is on the label, it may fade away. The printer quality should be good if we are using printed ones, and the format and the quadrant differs. Similarly, we have labeling requirements that are documented in the regulations for the cord cells and the stem cells. We have a 3-tier labeling system for the cord blood. It includes initial collection label that identifies the unit, the mother, the collection site, time, and carries the mandatory handling caution. Whereas at the second stage, that is during and following cryogenic cryopreservation, the core product identity label includes the name, volume, the cryoprotectant used, the dates, and the traceability. The further, before release, the issue label is attached, which includes the testing results, the clinical use safety statements for the receiving hospital. The testing details is extensive with respect to the cord blood or the stem cells, and it includes the TTI testing results and additional infection screening such as CMV or HTLV wherever it is applicable. In addition, it can include the total nuclear count, cell viability, blood group, sterility report, as well as HLA matching results. Finally, coming to the cellular therapy products, where India's regulatory framework is actively evolving, and this is the label content that travels along with the CAR-T units. On the container label, the product contents, the dosage are mentioned, and it's specifically mentioned that not for sale and for clinical trials only. The preservative present in the product, the shelf life, etc. And further, in addition to that, we have chain of custody record that is released along with the CAR T cell unit, which include the cell source, the batch ID, etc. So, with regard to the CAR T cell product, it comes under New Drug Clinical Trial Rule 2019, and the labeling requirements and the conditions of permission are laid down in the Chapter 8 of NDCT Rule 2019. Similarly, labeling requirement and the license conditions for the import for clinical trial products are mentioned in Chapter 9, and the importer identification details have to be there on the label. So when we move from clinical trial stage to the stage of implementation or the post-marketing phase, for example, the India's first indigenous CDSO approved CAR T cell product, that is NextCAR 19, is now in the market. There are a lot of changes that happens with the labeling in the clinical trial stage to the implemented stage. So this is one such example. Now, with this, I would like to conclude that regulatory framework already exists in India. However, the blood centers have implemented and all these mandated labeling standards. These are routinely assessed during licensing and inspection processes. Despite regulatory alignment, variability in practices persist, as we have seen earlier. So, closing these existing gaps and embracing the advanced technology is the way forward for safer, smarter, and globally aligned transfusion system. Thank you. And now I would like to invite our chairperson, Dr. Debashish Gupta, to take the sessions further.
Thank you, Dr. Shamee, for such a wonderful presentation on the labeling guidelines as is in practice in India. So now, good morning, everyone who are attending this webinar. And I would like to introduce our second speaker of the day, Dr. Joy Mammen. It's my pleasure to introduce Dr. Joy Mammen, who is a professor and head of the transfusion medicine at Christian Medical College, Vellore. And he has completed his undergraduate studies in medicine, and subsequently he specialized in pathology from the same institute. Dr. Mammen has been trained in laboratory informatics, and his area of interest includes proficiency testing, laboratory automation, voluntary blood donation, and public policy related to transfusion practices. Dr. Mammen coordinates 2 national ICWAs for hemostasis and transfusion medicine, and he is a member of the National Blood Transfusion Council of India. Dr. Mammen has served on the Institutional Ethics Committee for ICMR, NIRDH, and he consults for the World Health Organization on issues related to blood safety. Dr. Mammen has a lot of publication related to quality assurance, hemostasis, laboratory hematology, and transfusion medicine. I would like now to invite Dr. Joy Mammen to give his presentation.
Good morning, everybody. I hope I'm audible. I'd like to thank the organizers for this opportunity to share some of our experiences in incorporating ISBT 128 into our blood center and see how it can be moved forward in other organizations as well. I do not have any conflict of interest for this talk. I've served in the past on the board of directors of ICWA, and I advise certain startups but had no financial benefits from any of these organizations. So the question comes, why do we need to implement an ISBT 128 solution? And Dr. Shamee very clearly spoke to us about labeling being the first step of avoiding errors and implementing safety within the healthcare systems, and more specifically in terms of the blood safety issues. So for us, it was a natural choice. Once we had finished our digitalization and workflow automation, we wanted to stay aligned with standards in this concept of traceability and being able to ensure that your traceability is assured across multiple hospital information systems and lab information systems. And then our operational safety, of which I will discuss a couple of examples with. So our baseline scenario was that we were completely paper-based, and I think this is the state of many of the blood centers across the country today. A lot of the data is on paper, and so therefore it is challenging to start thinking about how to implement electronic systems. The challenge is also in terms of maintaining quality to make sure that all forms and registers are complete, avoiding duplicate entries. We sign in many locations to ensure that people take ownership for work. And then we all go through this process of making sure stock is refreshed and we do manual reconciliation. So digitalization seemed to be a natural answer to some of these questions when we looked at it from an organizational point of view. Just a brief mention about the data entry operator, which is the general way of how data is entered in a digitalization process. However, we have to come away from this and change to a process where each person is able to utilize the digital system or access the digital system at the point of interaction and upload data into it dynamically. So I've just put a short comparison here between the two. We won't go through the points, but essentially the traditional model of digitization is very challenging, and we should look at meaningful digitalization where access to compute is important and people interact with the systems as and when they are working, and it should be done synchronously and in a dynamic. Post digitalization, we managed to automate the entire workflow or digitalize the entire workflow. And then we realized that integrating barcode into this workflow became a logical part of this entire process. The other thing that we looked at this point was also interfacing our analyzers with bidirectional interface so that we could move data from the sample in terms of the label on the sample to the machine, and then the results would come from the analyzer direct. So these would reduce errors and avoid manual entry. And we worked on developing reports and registers which are required for our system from them. A brief shout out to the people who invented the barcodes. We are not familiar with these people. I think I salute them every day because given the scope at which we have implemented barcoding across systems, not only from our supermarkets, but also in healthcare systems, I think this has really reduced errors in the workflow and increased our efficiencies. Nowadays, there are alternatives like Bluetooth and RFID. But in a country which is very cost conscious, we should look for least expensive products that are available to us all. So key benefits, I think, are the error reduction because there's so much of manual entry. And each time you enter a patient's ID number or a donation ID number, especially with long meaning sequences of numbers, which are necessary in today's day and age. Sometimes they are concatenated numbers, and this becomes very challenging for human beings to enter and keep track. So in that sense, it is very efficient to have barcodes. And the second part is that it can be integrated with many other systems because these are machine readable and the data can be interchanged or exchanged between systems. And the two examples that I will discuss later are about bedside transfusion safety and operation room blood delivery. So how did we go about it? We became aware about the ISBT 128 through an attendance at the ABB way back a long time ago. We reached out to ICCBA to get the facility identification number. We worked with the IT team because we had an in-house project going on for the development of software for the blood center. And we went through the material available on the web, the documents and the presentations which are available. We signed up with ICCBA, and now on the member portal, thanks to the upgraded new website, you will be able to see a lot of information available already. And it is actually a much better site than it was previously. You can have access to a lot of information and health files already on the website. Plus, you can reach out to the team, some of whom are here already on today's meeting, to get better information about it. So who was involved in this? We had the clinician pathologist. I was the team lead for this. We had technologists, clerical staff, and we had programmers who worked with us. And then we worked with the companies from the analyzer and automation side. And the other important mention is the barcode label. As Dr. Shamee also mentioned, the physical characteristics and properties of the labels are also important. So we found a vendor who would fulfill all those requirements for us. So we had a phased implementation. We started in the same workflow that we usually use at each time, being a blood center with our usual demand for quality and being particularly focused on quality and error reduction. We had parallel systems for many things going on. Also, we had traditional paper-based systems and the computerized record, and then there was reconciliation of this. Then we went on to generate all the labels that are important within the system, the donor labels, the form labels, the donation ID generation, and all the linked issues that we used in our workflow. So the entire workflow needed to be mapped. I'm not going to go into details of each, but starting with the donor form and generating a donor ID, registering the donors, linking the samples to the paper forms, and then taking it through the process of collecting of blood, and then making sure that the barcodes were readable on the analyzers and within all the barcode readers within our systems. And then we also worked with the automation to make sure that the same barcodes could be read across the system. So finally, we end up with the ICCBBA ISBT 128 standard labeled product. This is a platelet product I think on this side, and on this side you have the red cell product. So the characteristics of the ISBT 128 standard label was mentioned. The important thing to remember here is that ISBT allows us to standardize our labels and make sure that the same information appears repeatedly every time we use it. There are certain minimum items that have to be specified: the donation ID, the group code, the expiry date, and the product code. And then beyond that, there is actually adequate space to put in all the requirements of the Indian licensing authority. Sometimes people think that we do not have space to put in what will we do with the regulatory authority. Actually, we have managed to put in all the requirements, which includes the name of the blood center, the license number, date of collection, the volume, the source, and all the instructions for use. And on the right side bottom column, we also have the TTI status and the additional information which has to go. So there is actually adequate real estate if you do the 100 into 100 mm label, and it should not be a problem at all for you to fulfill all the requirements. And now we know that there are ongoing conversations with CDAC Pune, which is the National Release Center for Healthcare Standards as well. So the question sometimes comes up: what happens if I'm using a commercial software? There are increasing number of blood center management programs. In India, it's an unregulated sector, so vendor to vendor, there will be substantial differences. You have to go through all the usual process of acquiring software, and importantly, speak early to your software vendor about the use of ISBT 128. If they're already registered, you're a happy customer. If not, please reach out to ICCBBA to have a discussion. And currently, they have an India Development Office. Dr. Shivanand, who's been in communication with you all, is the Indian office fellow. I will quickly go through in about 2 minutes about 2 use cases that we did in terms of vein-to-vein safety, where using the same ISBT 128 label on the final label on the bag, we generated what is called as the issue ID, which links the patient to that particular issue. And then this was matched up with the patient ID on their wristband, so that when the bag reached the bedside, they were able to scan it either with an Android phone or with a barcode linked to a laptop. And then if it was compatible, you would get a green signal. If it was incompatible or the wrong unit or the wrong patient, you would get an incompatible highlight. And then the application was able to trace the details. The second use case was when we started delivering blood to operation rooms using the pneumatic tube system. Traditionally, this is a big challenge because we have to have blood ready for use in operation rooms. And once it leaves our control, it's pretty challenging to manage these. So we designed it along with the biomedical engineers to transport blood and components to the operation room system. Initially, we validated the product itself and the time taken and the quality of the product when they receive it. And then we implemented the barcode-driven workflow. So the distance from our blood center on the ground floor to the last blood center up there, walking distance may be about almost 3/4 of a kilometer, but the chute system takes only a few minutes to deliver. So we have an electronic authorization system through which the blood is authorized for release. The blood is dispatched via pneumatic chute system, and it is scanned to receive and scanned to transfuse. All this is happening with the ISBT 128 standard label on the blood center, on the blood bag itself. So these are some of the visuals from that. In the blood center, we have a system that will display the request. The bag is identified, put into the pneumatic chute system. It's dispatched and received in the respective operation room. So I'll stop now. And the conclusion is that ISBT 128 offers a pathway to move forward. And as Dr. Shamee was mentioning, in a standardized manner. Blood mobility is rapidly increasing for different reasons. As we know, it is possible to move blood across blood centers. And the ability to have standardized traceability with identification of each unit is very important. ISBT 128 is now in India. And I'd like to thank the organization, my co-speakers in this meeting, and the team at Weber for this. Thank you.
Thank you, Dr. Mammen. Okay, now we are going to have a poll that you will be able to select an answer on. If we could display the poll. Okay, implementing the ISBT 128 standard in India. What is your current status regarding ISBT 128 standard implementation at your center? Your choices are not started yet, considering implementation, implementation in progress, fully implemented, or not applicable to my role or organization. Okay, and while we are waiting for the poll answers to come in, I just want to remind everybody that you can enter your questions into the Q&A section of the Zoom application. And if you cannot see the Q&A button, just select the More button, which is the 3 little dots, and the Q&A should be available to you. And I do see one question coming in. Can we get a recorded video? Yes, the recording of the video will be released in, I think we are usually around 2 weeks. Yes, I'm being told 2 weeks is when the recorded video will be posted on the ICCBBA website. Okay, and here's the answer to our poll. 41% said not started yet. 22% said considering implementation. 18% said implementation in progress. 8% said fully implemented. That's great to hear. And 10% said not applicable. So if I could go ahead, if we could close the poll and get our panelists to rejoin the stage so that we can have everybody answer questions, that would be wonderful. Thank you, Dr. Gupta.
Yes. So,
Dr. Chenna.
thank you for that wonderful session today. And now we can go for the question. There is one question on the chat box, which is being directed for Dr. Shamee. The question is, Dr. Shamee, what are the mandatory requirements that need to be on the label of an autologous unit?
Yeah. So mandatory label requirements for autologous units includes almost all the fields of the allogenic label, except for an important change that donor is recipient here. So there should be a statement for autologous use only, and this statement should be very prominently displayed on the label, mainly to prevent inadvertent issue of that unit to any other patient.
The second question that we have is, and this is open to anybody, what is your opinion on private agencies who also ensure safe vein-to-vein transfusions using barcodes, temperature regulators, etc.?
I think Dr. Shamee, you can answer this question.
Yeah, any such vendor, when we adopt, there are a lot of steps that we need to follow, mainly for validation of the newer system. We need to do a pilot study and validate the system before adopting, and then if it is fine, then we should definitely adopt.
Just I have an additional point, sir. Actually, there are a lot of vendors who wants to use the barcodes, but how many number of barcodes to be used and what information should be encoded in the barcode is not standardized, and that's where ISBT 128 comes into picture. ISBT 128 is not one of the vendors. It is just a standards body. It will guide the vendors and software people like how to make best use of the barcodes and the other advanced technologies.
Thank you. Chenna, would you like to read the next question?
Yes, yes. Yeah, so the next question is to Dr. Joy Mammen, sir. So it is by John Wesley. He is asking whether does the standard help in blood transfers between blood banks both within the same organization? He has given an example like the town campus, Ranipet campus, and outside the organization.
Yeah. So we actually have 5 blood centers within the same organization and 5 different remote locations in our community hospitals, and also 2 other blood centers which are organizations which are not part of our institutions. And in both cases, within our organization, it is completely integrated. So we can transfer stock across centers, and the same identity is managed across the centers, and people use the same identity system. When we send blood to our to the storage centers which we're supporting outside of our institution, we still use the same barcodes, and we instruct our partners who are using it not to stick any other label above the barcode label, because that is the final compliant label and they have to use that. And so in case they return it as stock which is unused for refresh, we still have the same label. Some of them choose to use only the barcode number on it, and they have developed their own hospital information systems which will accept those barcodes for scanning and use within the hospital. So once you have a barcode on it, it's useful, and it can be managed if the IT team is also collaborating with the blood centers.
Dr. Gupta, would you like to ask the next question?
What are the pros and cons of implementing the ISBT 128 standard? I think that is this question is very much interested for all the delegates who are attending this webinar. So we have to know what are the pros and cons. So anybody, either Robin or Kayla, can you give a little bit of your experience in this thing?
I think Dr. Mammen, did you want to say something? You had your hand up. Sorry.
I'll just be brief because I already presented. I'm not going to talk much more. But certainly the advantages outweigh the disadvantages, and the whole country and the health system within India is moving towards standards and using international standards. We have already declared that SNOMED, CT, and ICD are the international codes. LOIN, for example, for the lab identifiers are the accepted code. So there is no reason why we should invent our own coding system when it comes to blood and tissues and other bio compatible materials or materials of human origin, medical products of human origin. So in terms of a very basic understanding, I think using standards and going forward with aligning ourselves with international standards is. I'll stop over.
Thank
One
you,
more
Dr.
comment I just want to make is that the involvement of the National Blood Transfusion Council and the Drug Controller General of India should be involved in this particular ISBT standard implementation, because the guidelines has to be created so that all the blood, because India has more than 4,200 blood centers across the country, and it will be a Herculean task to implement the ISBT 128 standards to all the blood centers unless until the government come into the picture and they develop some guidelines in how to go ahead and instruct the centers how to implement it. Yeah.
Thank you, Dr. Gupta. Dr. Chenna, would you like to ask the next question?
Yeah. So the next question is by Bucky Israel Karuna. He wants to know what is the significance of the number in box written vertically in top right corner of ISBT 128 labeling.
If I'm not mistaken, the character written in the box that is next to the DIN is actually a check character, so that when you're typing in the donation identification number, the DIN, into your system manually, when you type that check character, or if you visually check the check character, the computer system should let you know, yes, what you typed into the system is actually a valid sequence.
So the next
And
question, what is the challenge with respect to data migration while changing the software?
okay. So Dr. Chenna, would you like to? Okay, so
Yeah.
oh, what is the challenge from? I see. I apologize. What is the challenge with respect to data migration while changing from one software system to another blood center? And do you think standardized data can solve many of those issues?
Yes.
Go
Definitely,
ahead.
standardizing will help in data migration because the traceability from the donor to the patient and look back plays a major role, and hence it is very important to have the data migration efficiently done.
Okay, the next question is for Dr. Gupta. In India, many blood centers have already started using barcodes and RFID. So, do you think it is the right time for regulatory authorities to formulate guidelines regarding how to use such electronic information exchange methods effectively? And if yes, where do we start to bring out such Indian national guidelines?
Yes, I fully agree that it is the right time for the regulatory authorities to formulate guidelines regarding the use of such electronic information. And already being a member of the National Blood Transfusion Council, as well as I am also part of the disaster management, where the transfer of the blood is one of the very critical issues from one aid to the disaster site. We are already trying to incorporate this type of technology in our guidelines. Okay, but it will take some time where we can fully, because there's a lot of issues in the country regarding the existing infrastructure facilities, the manpower, internet access. All these things need to be addressed before we go on a full-fledged of electronic implementation.
Thank you. Dr. Mammen, how does ISBT 128 improve component tracking in resource poor or remote settings?
Okay, thank you for that question. I think there are two parts to this question. One is to ensure access to blood and blood components in remote areas is a challenge. And we know that the country's policy is to actually allow the blood storage units or the blood storage centers to be the channel which are linked to mother blood banks, which will provide and refresh stocks. Part of the challenge of providing blood to the remote centers is that you're not able to track the unit once it leaves the blood center. So whether it was transported using cold chain and whether it was maintained as stock in the remote place using cold chain, did they have continuous supply of power to ensure that the cold chain was completely maintained, so that the main mother blood bank can take back a new stock and refresh it. Earlier on, I think in several states, when we started this program, there was a report of high level of wastages in India also, in some of our states, because the blood would leave the mother blood center, go out to the peripheral blood bank, blood center, the storage unit, and then we had no idea how to track the temperature over there and whether the cold chain was maintained. So the mother blood bank did not want to take it back. Today, fortunately, there are good technologies available, and where ISBT plugs in, 128 standard plugs in, is the fact that you have a unique, identifiable, traceable number on every unit that you're moving out from. You can use it either as a barcode or as an RFID tag. Both standards are available now. And so therefore you can track the unit. And there are also companies within the country which provide cold chain equipment and cold chain tracking systems so that you can manage to track the inventory unit from your blood center to the periphery throughout the life of the unit there and back to supply, back to the mother blood bank if it needs to come back. So there are like end-to-end tracking solutions which are available. And so therefore, I think it is possible to supply good components, good quality components wherever it is required. Thank you.
I think there's
Robin,
one
I have
question more for, before we add two more questions, I think we can, one is for Dr. Joy, is that though there are very less number of cord blood centers operating in the country, but it is known that the ISBT 128 labeling is mandatory. Is it a fact, or is it they are practicing on the
I
cord blood
am not
centers?
sure of the answer for that because we don't run a cord blood center here, but can either Shiva or somebody from Dr. Shamee, would you like to tackle the question? I'm not aware of the answer for
Sir,
that, but I
The
can get back
very
to
less
you.
number of blood centers in the country.
Yes, sir. Actually, in India, there are a lot of centers registered with ISBT 128, and many of them are cord bloods. And according to Indian guidelines, they recommend following the AABB guidelines for the cord blood. And indirectly, the AABB mentions ISBT 128 to be mandatory. So, because of that reason, I think cord blood centers are using ISBT 128 labeling in India, sir. Anyway, I'll just confirm that fact again. As per my understanding, most of the cord blood centers are already using it in India.
Can I add
Thank
a
you, Dr.
point to the previous answer? Is that in ISBT 128, there is a provision to provide pre-printed labels also. It should be used carefully. You still need some IT system support. So you should speak to your vendor who can link up with ICCBA and find out how you can use pre-printed labels. So you don't have to be the one producing barcode labels, but you can still integrate it into your system. There are ways to do that.
Yeah, I have a question for Robin. What is the
Yes.
purpose of the flag character in the DIN?
Okay, so the flag character are the two numbers that are rotated on their side in that in the sequence after the DIN and before the check character. The flag characters can be used for process control. For example, in the blood bank that I worked in, we would put a certain flag number on the donation record, the actual piece of paper, and then each test tube that would go to the testing lab that was accompanied the collection would each have its own flag character as well. And that way, a lot of testing facilities can use those flag characters to determine what that test tube is. Because once you take the top off of a tube, how do you know what the type of tube is? So they're very useful in process control. Okay, should we move to Dr. Shiva's closing remarks? I want to thank everybody for being here today and really appreciate you all and your wisdom. So thank you very much.
Yeah, good day, everyone. So as we come to the end of this enriching webinar, it's my privilege to deliver the vote of thanks. On behalf of the organizing team, I would like to express our sincere gratitude to our esteemed chairpersons, Dr. Debasish Gupta sir, Professor and Head, IHPT NIMS Jaipur and also President of ISTM, and Dr. Deepika Channa Madam, the Additional Professor, IHBT KMC Manipal, for their valuable guidance and support throughout the program. My heartfelt thanks to our distinguished speakers, Dr. Joy Mammen, Professor and Head IHBT CMC Vellore, and Dr. Shamee Shastry, Professor IHBT KMC Manipal, for sharing their expertise, insights, and experiences. Your presentations have greatly enriched our understanding and made this session both informative and engaging. I would also like to acknowledge Dr. Ganesh Mohan sir, Professor and Head IHBT KMC Manipal, and Mr. Owen McGrath, Executive Director of ICCBA, for their invaluable support in making this webinar possible. And a special note of appreciation goes to Mrs. Robin White, Technical Director ICCBA, for expertly moderating the session. And we're equally grateful to the dedicated ICCBA team working behind the scenes, Kayla, Beatrice, Chris, and all other ICCBA staff members, for their meticulous planning, support, and seamless coordination. Last but not the least, I extend my sincere thanks to all the participants. Your enthusiastic participation and excellent turnout have been the true driving force behind the success of this webinar. Before we conclude, a small reminder that the participation certificates will be sent to all the attendees within approximately 2 weeks. Once again, thank you all for your time and support. We hope you found this webinar valuable and beneficial. Stay safe, stay connected, and have a wonderful day ahead. Thank you.